Good morning everyone. Today we are going to discuss about our last module, bioinorganic chemistry. So in this module, actually we are dealing with the role of various elements in biological system. So let's begin. So bioinorganic chemistry is an interdisciplinary scientific branch that examines the chemistry of compounds within biological system. So these elements are generally divided into essential elements and non-essential elements. So it is possible to divide the 30 elements present in biological systems into two. First one, essential elements, and second one is non-essential. So the essential elements, from the term itself we can understand they are very much required for the daily activity of an organism. So these are elements which are essential for the maintenance of life in organism. Essential elements can be further divided into two. That is bulk or macro elements and trace or micro elements. The bulk or macro elements, from the term itself we can understand they are required in large quantities by living organism. Examples are oxygen, carbon, hydrogen, nitrogen, sulfur, sodium, potassium and magnesium. And the trace or micro elements, these are required in small quantities by living organisms. Example, copper, zinc, cobalt, manganese, etc. And now we have another class called non-essential elements. These elements which are not actually non-essential, but their function in the body is not yet known. That's the thing, that's the problem. So in our body we can find these elements, but their functions are not so well known. So those elements are considered as non-essential. Examples are bromine, boron, silicon and nickel. Then metals in biological system. So in the previous session we discussed about various elements in biological system. Similarly, we can find metals in biological system. Some of the most important functions of metals in biological systems are: number one, as cofactors in enzymes. We know that enzyme is actually consists of a proteinaceous part and a non-proteinaceous part, and the non-proteinaceous part may be a metal ion. Okay, so that's called a cofactor. So cofactors are important for the catalytic activity of enzymes. Metals like Mg2+, Ca2+, zinc and manganese ion, etc. can act as cofactors. An enzyme that has tightly bound metal ion is called a metalloenzyme. See, the cofactor may be tightly bounded with an enzyme, and such an enzymes are called a metalloenzyme. Then similarly, there will be very weak interaction between the non-proteinaceous part with the proteinaceous part, and we will discuss them about later. So if a metal ion that has a very strong bond with the enzyme, it is called a metalloenzyme. A metalloenzyme, keep in mind. The metal ion may contribute to the structure and catalytic activity. Example, alcohol dehydrogenase contain a Zn2+ ion. So it's a zinc containing enzyme. Urease containing Ni2+. Carboxypeptidase containing Zn2+ are examples for metalloenzymes. So keep in mind some examples of metalloenzymes. An enzyme that has a loose bound metal ion is called a metal activated enzyme. Suppose the metal ion may be loosely bounded with the enzyme, the proteinaceous part, then we call it as metal activated enzyme. Metal activated enzyme. Here the presence of activator metal is required for the enzyme to show catalytic activity. Example, phosphofructokinase. Phosphofructokinase. Then second one, the first application or the first use of metal ions is as a cofactor. Second one as structural entities, because we know that in bones and teeth we have calcium. Similarly, in order to provide a structure to organism, it requires certain metal ions. So in the control of, third one, in the control of metabolic mechanisms, metal ions play important role in control of metabolic pathways. Copper is important for the proper protein metabolism in organisms. So it is important for the digestion of protein. Then manganese is important in protein metabolism and also controls oxygen release by photosynthesis. Calcium along with the magnesium controls muscle contraction. So these are the some other examples of metal ions involved in biological system and they control metabolic mechanisms. Then the fourth one, it's very familiar as oxygen carriers. We know that in human beings, hemoglobin is the oxygen carrier. So there is a metal in hemoglobin called the iron, and along with iron, the hemoglobin performs its duty. Then fifth one, maintenance of osmotic pressure and pH. So you might be aware of sodium potassium pump. So it's actually a way to maintain osmotic pressure. So sodium, potassium, calcium and magnesium maintain the precise gradient across cell membranes and thereby maintaining the osmotic pressure and pH. Some examples are sodium, potassium, magnesium, calcium, etc. So they regulates a osmotic pressure. Keep in mind, and just read down all those things. It's just an example. So more examples are here: chromium, manganese, cobalt, copper, iron, etc. We will discuss about them in detail. First of all, biochemistry of iron. Okay, we have to discuss about some major metal ions involved in biological system. First one, iron. So biochemistry of iron. Iron is essential in small amounts for both plant and animals. However, it is toxic in large quantity. Iron acts as oxygen carriers in the blood of mammals, birds and fish. Okay, and hemoglobin is the what? Hemoglobin is responsible for the carrying of iron. Okay, so for oxygen storage in muscle tissue, we have myoglobin. So the function of hemoglobin is oxygen transportation, whereas that of myoglobin is storage of oxygen in muscles. Then as an electron carrier in plants, animals and bacteria for electron transfer in plants and bacteria. So you might heard about certain mechanisms in plants, like there are several what we call like Krebs cycle, etc. We are aware of such kind of cycles in organism. In these cycles, the iron is involved, and it actually carries certain electron and that transfer to some other species and perform certain reaction. So that's why it is called as an electron carrier in plants, animals, bacteria, and for electron transfer in plants and bacteria. So in Krebs cycle, you can find there is a transfer of electron, NADPH to NADP, etc. And during such kind of transformation, iron is involved, and during the transformation, iron actually donates its electrons. Then we have ferritin, transferrin and hemosiderin. Helps for the storage and scavenging of iron in animals. Then iron is an important part of nitrogenase enzyme, which is found in nitrogen fixing bacteria. So in all these things, iron atoms are involved. Okay, along with the transfer as well as storage of oxygen, iron has a lot of roles in biological system. The majority of the role is carrying and storage of oxygen. Along with that, it performs a lot of duties. The protein hemoglobin serves for the transportation of oxygen, and protein myoglobin serves for the storage of oxygen, which are essential for the life functions of all vertebrates. The active site in both proteins is the planar heme group. So in hemoglobin, there is a heme group. Then what is that heme group? A heme group consists of an iron atom which is coordinated to 4 nitrogen atoms of porphyrin ring, and it is embedded convoluted protein chain convoluted protein chain. I will show you the picturization, then you can easily understand. The heme group is attached to protein chain through a nitrogen atom of histidine. Histidine is actually an amino acid, and this is how it looks like. And this is porphyrin. So in the center of porphyrin you can find a iron atom, and together they constitute a heme group. So a porphyrin ring, and at the center, if there is a iron atom, they together constitute a heme group. Okay, so it's actually the proteinaceous part. Then you will find it requires a histidine atom, and I will show you that here. See, these four nitrogen atoms are actually a part of a porphyrin ring, and here the iron is in a 6 coordinated state. Now the 4 coordinations are occupied by the nitrogen of porphyrin ring, and the remaining 2 is occupied by either water or oxygen, and the remaining 6th one is occupied by nitrogen of histidine. So this is how the 6 valency of iron occupied in hemoglobin. So as we said earlier, one of the valency is occupied by either water or oxygen. It's not fixed. It is occupied according to their concentration. If water is in high concentration, then definitely water can occupy, and if there is a site where if there is a very good amount of oxygen, then oxygen will be occupied. Okay, and this is how it happens. It is an equilibrium reaction based on Le Chatelier's principle. Greater the concentration, greater will be the forward reaction. Similarly, greater the concentration of water will be occupied at the one of the position of iron, and if there is a very good amount of oxygen concentration, then oxygen will be occupied. Okay, so this is how a hemoglobin looks like. That's a real term. Okay, then the myoglobin contain one heme group, and its molecular mass is 17,000 AMU or unified mass. 17,000 unified mass. It binds to oxygen more strongly than hemoglobin. That's why myoglobin can actually store oxygen. So the bond is more strong when compared to that is present in hemoglobin. So the oxygenated myoglobin is called oxymyoglobin. Hemoglobin contain 4 subunits, each contain heme group. So actually it consists of 4 heme groups. Since hemoglobin consists of 4 subunits, each contain heme group. So definitely in 4 subunits there will be 4 heme groups. Each bind to one oxygen molecule. Molecular mass of hemoglobin is 64,500 unified mass. The oxygenated hemoglobin is called oxyhemoglobin. The 4 subunits are linked with one another through salt bridges present between 4 peptide chains. You don't need to remind all those things. Just for information, keep in mind. That's not so important. The important thing is, what is a hemoglobin consist of? It actually consists of a porphyrin ring. At the center there will be an iron in +2 oxidation state, and together they give a heme group. Okay, so in hemoglobin there are 4 subunits, means there are 4 heme groups. Then the amino acid sequence in both hemoglobin and myoglobin are different, but their tertiary structures are similar. Actually they are proteins. So they have primary, secondary, tertiary structures, but their tertiary structures are similar. The oxygen transport of hemoglobin is manifested in 2 ways. The Fe atom binds to oxygen molecule and transport them from lungs to muscles, where they are delivered to myoglobin molecules, and the hemoglobin then binds to carbon dioxide from the cell sites, from the muscle sites, to carry it back to lungs. So this is how the mechanism works. the oxygen, carbon dioxide transportation, how this works. The 5Fe2 plus is present in deoxyhemoglobin and deoxymyoglobin lies about 0.7 to 0.8 angstrom above the plane of porphyrin ring. So this is actually the porphyrin ring. So there is an upper gap as well as there is a lower gap. So those gaps are described here and you don't need to worry about all those things, not so important. The iron is coordinated to 4 nitrogen atom of the porphyrin ring and the nitrogen of histidine group. Then in deoxyhemoglobin and deoxymyoglobin, the position trans to histidine group is occupied by water molecule, but in oxyhemoglobin and oxymyoglobin, the position is occupied by oxygen molecule. Keep in mind, this is very important. This point is very important. In deoxyhemoglobin and deoxymyoglobin, the position trans to histidine. Trans to histidine means this position, it is occupied by water molecule. But in oxyhemoglobin and oxymyoglobin, it is occupied by oxygen molecule. We may think in the absence of oxygen, this 6th position may be occupied by carbon dioxide, but it is actually occupied by water molecule. Okay, now we will discuss about the oxygen mechanism, the mechanism of oxygen binding, that will be discussed later. That's all about today. Thank you. We will discuss about the rest in detail in next class.